cases have appeared in New York City. So we'll see what happens with all of this. It seems to be here. So just be cognizant, wash your hands, everything we discussed on Friday, okay? And I'll keep y'all updated on these numbers as we go through the semester. So today, I want to finish up the lecture on adhesions and proteases. We'll go through the case study, and we're going to work on a new case study today, the biofilms case study. Hopefully y'all all got a chance to look at this over the weekend. I put it all online for you, and I mentioned it on Friday. So I gave y'all all handouts with multiple copies of the case study. Most groups have individual case studies, so they're all a little different from other groups, and it's supposed to represent the different branches that would be involved in a bioterrorism incident and how those entities actually respond to these cases. Okay? Graduate students, send me your pre-proposals today. If you want me to look at them, I will get them back to you in the coming days. What we don't get done this week with this bioterrorism case study, we will do the Monday after spring break. So I'm sure y'all are all fully aware that next week is spring break. I won't be here, y'all shouldn't be here. Enjoy it, and when we come back that Monday, we're going to finish up the bioterrorism case study. We're going to do a review for the test that's going to be on Wednesday, okay? All right. Any questions over that? Anything that we got going on? Okay, let's start out with the case study from last week. Okay, so this was about what's the molecule of inheritance. Y'all should have all read through this case study. Strep pneumoniae was important in the 1920s, this first question, because they were a major cause of death before the age of antibiotics. Okay, so before antibiotics came into circulation, strep pneumoniae was a big problem. All right, so going through this, when you answered on your handouts, what did y'all predict was going to happen for each of these? Okay, so with the rough strain, from what they told us, this rough strain was not supposed to kill the mouse when it was infected with this rough strain. The smooth strain, they knew did kill the mouse. The heat-killed smooth strain, once you kill this bacteria, it does not, no longer is virulent in the mouse, it doesn't kill the mouse. So what did y'all predict right here with the rough strain and the heat-killed smooth strain? Anybody? Yeah.
The mouse would die.
The mouse would die. Okay. Yeah. So would you predict that based on these results? So if you infected with the rough strain, the mouse would live, right? And with the heat-killed smooth strain, the mouse would also live. So I would have predicted the mouse would live. But yeah, I get it. You know the case study. And of course, that's what happened in the second part. Yeah. So the actual results of the experiment, the mouse does die. Okay. So which of these treatments were controls?
First two. The first two.
Three?
First two.
First two? Okay, why do y'all say two?
Because nothing's happened for you considering the master out.
Because nothing's happening?
Yeah.
It was treating them with.
Okay, okay. Yeah, so why would you think that this third one is not a control? Okay. So they modified the bacteria and they wanted to know what happens. But they did know the outcome. So actually these first three are considered controls because they did know the outcome. So,
If it had like a capsule, right, wouldn't it also, if you heat killed it, wouldn't the capsule still be toxic to the mouse and potentially cause it to die?
it could be, yeah, in this case it's not.
Okay.
So, yeah, with some capsules, it could still stimulate an immune response and cause an issue, for sure. Okay, but they did know the results from these first three, so that's why they're considered controls, just FYI. Okay, which of these results was unexpected? Is what we already discussed, the last one. What are two hypotheses that might explain the unexpected results? Did y'all have any hypotheses that maybe weren't transfer of DNA? Did anybody think of anything else here? No. Like maybe the capsule from one could have been acquired from the other one and caused it to be virulent? No, nothing? Nothing else? Okay, well, so you all know the answer. You all have been through this, of course. So the hypothesis would be that there was transfer of genetic material from the heat-killed smooth strain to the rough strain that allowed it to build this capsule. Okay, enzymes as tools. So these are definitions that I want you to know: proteases, DNases, RNases. Know this for the test, okay? Know what each of these are. Yeah, and you would expect each of these to catalyze a certain reaction based on its name. Proteases degrade proteins, DNases degrade DNA, RNases degrade RNA. Okay. And so, how could you treat the dead smooth bacteria with these enzymes to determine whether DNA and RNA or protein is the united material? Of course, you do this individually, where you take a heat-killed smooth strain and you treat it with each of these enzymes, and the one that causes the mouse to live is likely indicative of the material that's required for the Okay. And so that's what they did. And the conclusion drawn from these data are that DNA is and so this was a very basic case study, but I want y'all to get the definitions out of this. Proteases, DNases, RNases, know what those are responsible for. Okay? Alright. Any questions over this case study? Okay. The one that you have in front of you right now is much more in-depth. Okay? So be prepared for that. So going to the left here. So proteases is what we're going to talk about for the first 10, 15 minutes of this course, or this lecture. Proteases are enzymes that catalyze the cleavage of peptide bonds, and they're very specific to their target proteins. Proteases are degraded enzymes. They're highly specific. They have highly specific targets in cells. The major uses are zymogen activation. So a zymogen is a protein that needs to be catalyzed, or it needs to be processed by a protease to become active. Okay, so it's an enzyme in its inactive state, and once it's processed by this protease, it becomes active. Blood clotting. We're going to talk about a case where proteases are important in blood clotting. Processing and transport of secretory proteins across membranes. I'm going to mention one case of this as well. Alright, so protease classifications, exopeptidases, based on the name, we should expect that this catalyzes cleavage of the terminal peptide bond to release a single amino acid. So it only cleaves the peptide bond at the end of the protein. There are aminopeptidases that cleave the N terminus of a protein. Here's an example, aminopeptidase 1 in E. coli. Carboxypeptidases cleave peptides at the C terminus. Okay. Endopeptidases break bonds of non-terminal amino acids. So these peptidases cannot break peptides into individual monomers. Okay? So be aware of those differences. These are typically very, very specific. Endopeptidases, there's a couple different classes, and these are really classified based on their active site residues. So what they use to coordinate a ligand in their active site to induce cleavage. Okay, so there are serine proteases. These rely on a serine residue in the active site, and these are mostly secreted as autotransporters, so through the type 5 secretion system. Okay. There are cysteine proteases. ESBL is secreted by EHEC to cleave receptor interacting proteins, which aid in post-inflammatory signaling during infection. So this is another mechanism that EHEC uses to subvert the post-immune system. There are Spartic proteases, which are dependent on active site aspartate. These are rare in bacteria. They're used for other enzymatic processes such as milk clotting or cheese manufacturing. There are also metalloproteases that are dependent on divalent cations that sit and are used to coordinate peptides in the active site of these proteases and to induce healing. Okay? So just know that these are the different types that you're going to need to know for this class. So an example of a protease being required for infection, there's a protease in HEHEC that actually cleaves this one protein in this core complex, this ESPB protein, as you see here. So it's naturally cleaved by a protease that's secreted by HEHEC, and this enables it to fold better and stick into the membrane of the eukaryotic cell when it builds this type 3 secretion system. Okay? What this group found, this is actually a group over at UT Southwestern that found this, is that Bacteroides theta iota omicron, which is a bacterium that we talked about, it's a natural part of your microflora, your gut microflora, actually produces these proteases that also cleave SB. So, when EHEC is in the context of an infection in your gut, the microflora is also producing these proteases, and so they're actually inducing an infection, okay, dependent on these proteases. And as you can see here, when it's only EHEC, this SB is not effectively cleaved, but when B theta is actually co-incubated with EHEC, you see this cleavage product right here. This is the processed form of SB that helps it to fold in the membrane to form this core complex, okay? So there is some indication that our natural host microflora is actually helping some pathogens with infection, ultimately. All right? So be familiar with this example.
So where's the protease?
So the protease would be in the extracellular environment right here.
Okay.
So
Which part?
right here in the extracellular environment. So this is the bacterial cytoplasm, the inner membrane, glycan cell wall, the outer membrane, and EHEC, as you know, builds this type 3 secretion system, and it directly attaches it to a eukaryotic cell. And so this would be the eukaryotic cytoplasm. This would be the host membrane right here, and SB is one of the proteins right here that forms this core that allows EHEC to translocate effectors into the host cell, okay? So the proteases would be here in the extracellular space. They're secreted by both EHEC and B theta, and they secrete this SB protein when it's being transported and integrated into the membrane of the eukaryotic cell. Okay? Any other questions? Okay. Additionally, I uploaded a paper for y'all to look at for your reading pleasure, scientific reading pleasure. A role for proteases in wound healing is what the paper addresses. So cutaneous wound healing reforms the skin integrity. So when you get a cut or a scrape, your natural immune system and this wound healing process works to reform the skin that's been damaged. Okay. So it produces the scar tissue, and ultimately these matrix metalloproteins, which you produce, control the degradation of the extracellular matrix during wound healing. So any misregulation of these NMPs can be detrimental for effective and efficient wound healing. Okay. Too many of these matrix metalloproteins can lead to destruction of the wound extracellular matrix. Bacterial pathogens, in addition to these MMPs, when they're misregulated, they can be detrimental. Bacterial pathogens produce proteases that can delay wound healing by degrading fibroblasts and immune cell mediators. This is done by not only secreting specific proteases, by also upregulating production and activity of these matrix metalloproteins, okay? So there are two ways that bacterial infections, skin infections, are detrimental to wound healing. Pseudomonas aeruginosa, which we're going to talk about a little bit here in a minute, is a human pathogen that produces proteases to limit wound healing. It also misregulates the activity of these matrix metalloproteins. Okay? So Pseudomonas is a big problem with skin infections, as well as other sites on the human body. It's been most associated with cystic fibrosis patients, causes big problems with cystic fibrosis patients, but it also plays a role in wound healing. And when you get a Pseudomonas infection on an open wound like this, it can be very, very, very hard to treat. And it can be very, very detrimental to the patient. It often infects chronic wounds and forms biofilms. And we're going to talk a little bit about biofilms. And these biofilms are structured communities of microbes that network together in order to adapt to adverse conditions. Right. So these biofilms are extremely difficult infections to treat with conventional antibiotics. So why is that? So this is the biofilm life cycle that Pseudomonas aeruginosa undergoes. So it can be a single free-floating bacterium. And when it lands on a surface, such as a wound, it can start to, these cells start to replicate and aggregate and attach to each other. And soon they further grow, and they start to form this extracellular matrices that are indicative of these biofilms. And then you get these mature biofilms that form. These biofilms are very, very difficult to treat because antibiotics cannot penetrate these biofilms. So there's a diffusion gradient, and the extracellular matrices that form these biofilms are composed of sugars, proteins, DNA, all sorts of different molecules. And when you treat these with antibiotics, they cannot diffuse into the center of these biofilms to effectively kill bacterial cells. So you might get some level of antibiotic permeating the very surface of this biofilm, but there's no way that they can penetrate deeply into these biofilms, okay? So it's really hard to eradicate these biofilms once they've formed in an open wound. Another part of this life cycle, this biofilm life cycle, is ultimately these cells.
That's actually really interesting because usually in patients that get acquired infections from being treated in hospital settings, it's usually because they're immunocompromised and because of this bacteria. They also like to attack, they don't attack healthy people, they attack HIV patients and cancer patients.
Yeah. So Pseudomonas is found all over the place. And if you are immunosuppressed, it can be very detrimental. And so that's why hospital-acquired infections are so bad. There are several of these bacteria that we're going to talk about later in the semester that run rampant throughout hospitals, and they really affect the outcome of these hospitalized patients with compromised immune systems. And they're known for their hospital-acquired infections, and that's why they're so problematic.
I was just asking, is it so resistant against antibiotics because one of the places it cultivates is in hospital settings, or is it antibiotic resistant because it cultivates there?
Because it cultivates in the hospital setting?
Yeah.
So I think that it's resistant to antibiotics because it forms these biofilms.
Okay.
There are other mechanisms of antibiotic resistance that can definitely contribute, but most Pseudomonas individual cells do form these biofilms, and these alone are very resistant to antimicrobial treatments.
Isn't this a huge problem, like burn wards, because you don't have enough skin?
Absolutely, yes. Heather?
So, we're talking about how, I mean, there must be some way to treat these patients. So what do? Just blast them with really strong antibiotics?
So yeah, they blast them with really strong antibiotics, topical antibiotics. There are new treatments where you're using magnetic waves actually disrupt these biofilms and then treat them with antibiotics. So essentially what you need to do to treat this is you need to mechanically disrupt this biofilm. So you need to break it apart so that the antibiotics can actually penetrate into the biofilm and kill the bacterial cells. So there's lots of different labs that are approaching that problem in different ways. But ultimately you want some mechanical disruption. And so a lot of times with skin wounds, they will go in and physically disrupt the biofilms that are in there and then treat with antibiotics. So,
What is the composition of the biofilm itself? Is it more like a mucosal surface, or is it kind of like
a thick, sticky matrix. So it's just DNA, which is very thick, complex carbohydrates, very thick, lots of cells, lots of proteins, just all sorts of junk in there. Yeah.
So how is it DNA? They secrete DNA just to.
Yeah, they secrete DNA. Also, when cells lyse, there's free-floating DNA in the environment, and it just gets a clump of DNA. It makes it really difficult to penetrate.
Are these
Oh, go ahead.
are these the same bacteria that's formed in plaque, like in causing gum disease and
Say
stuff?
that again, I did not hear.
Are these the same bacteria that cause gum disease and found in plaque in our mouth?
Not so much, no. But those are also biofilms. So your pathogens in your mouth, they also form biofilms on your teeth and gums and cause all sorts of issues there, for sure.
In my experience, when a patient comes in with this type of infection, the provider will take tissue samples or blood samples, whatever organ is infected, they'll send it off to the lab and they'll cultivate a media trying to test which antibiotics it's resistant against, and the ones that it finds that it's not resistant to, they'll usually pair it with two on it and then try to attack it.
So antibiotic cocktails, yeah.
Yeah.
We're losing the effectiveness of many of our antibiotics. So now they are using many antibiotic cocktails where there's a synergistic effect between two or multiple antibiotics to actually treat stuff like this. And yeah, that's a big problem with antibiotic resistance. And we're gonna talk about it in test 3, for sure. Okay, anything else? Okay, yeah, but just know that these biofilms are very problematic for infections. And lots of bacteria do this. Bacteria pathogens in your mouth, bacterial pathogens that affect the skin, cystic fibrosis patients, they get these large biofilms in the lungs. It's very problematic, and it's hard to treat these patients, or hard to treat the infections in these patients. Okay, so so in the case of Pseudomonas, biofilms are extremely detrimental to wound healing. They produce these proteases that inhibit wound healing. These proteases are called LAS A and LAS B. They are secreted from Pseudomonas via the type 2 secretion system. So are these proteases secreted in a folded or unfolded form? Anybody?
They're folded.
They're folded, yes, they are folded. Okay, so just be aware of that. Hint, you might say that again. So they're secreted in response to chemical cues from the host, okay? So during an infection, these things, the genes encoding these proteins are upregulated and they're secreted by this type 2 secretion system to help promote Pseudomonas establishment in an infection. So elastase, which are these proteins, LAS A and LAS B, these elastases and other proteases degrade elastin, which works with collagen to form the mechanical properties of connective tissue. So when you get these bacterial infections, your wound will never heal, okay, unless there's antibiotic treatment to remove this pathogen from the wound.
What's the why is like the meaning of the importance of the proteins being folded?
So what's the importance of the proteins being folded?
Yeah, that's
So so why is it important that they're folded? So I asked you, so what I want you to know is that there are different secretion systems that bacteria use to secrete these proteins, and via the type 2 secretion system, they are folded proteins. So So that's just what I want you to know. We've already gone over that. So I just want that to be a reminder that there are different secretory systems. As far as the importance of them being folded intracellularly before they actually exit from the cell, is that they probably will not fold properly once they're in the extracellular environment. So there are probably chaperones in the bacterial cell that aid in folding of lattice A and lattice B so that they're actually enzymatically active proteases, okay? And then once they leave the cellular environment, they're, if they were to leave unfolded, they wouldn't be able to properly fold.
And you mean that that's important because those could be targets?
Yeah, absolutely.
For antibiotic treatment.
Yeah, so if you can't secrete a folded protein, it could help prevent disease.
Are there antibiotics that target those systems like that?
I'm not aware of any, no.
Is that just something a little bit further on that's maybe a little bit ahead of where the field is?
Yeah, we're not there yet. So most of the antibiotics now target essential processes in bacteria. Very few of them target a process that is required for virulence but isn't required for cell survival. So the strategy now is just to kill everything, is to kill all the bacteria. However, more targeted antimicrobial therapeutics, narrow spectrum antibiotics that just target things that are required for virulence could be a very useful strategy because it doesn't add the same selective pressure for these cells to survive, so these cells aren't needing to come up with antibiotic resistance mechanisms. Okay. Does that make sense? Yeah, but we're not there yet. I mean, there are inhibitors that are being looked at, but there are definitely none that are on the market right now. They just target the process, one process necessary for virulence.
Yeah, it's just curious, I know it's quite a bit out of the scope, but like, is that just because of the conserved nature of a lot of the protein structures and how they might be similar to certain proteins that we have? It could.
I just think they haven't gotten there yet. Yeah, I see what you're saying, that there could be some conservation of proteins, they could be more broad spectrum.
Which it could affect ourselves.
Yeah, so for sure, I mean, absolutely. There could be some overlap in toxicity like that. But it's things that are, these ideas for new antibiotic treatment methods are out there, but nothing's been actually done on this stuff. So if y'all read through the paper that I put in there online on campus, I think I put it on Friday, it should be under today's date. If y'all read through, it talks to you about the different stages in wound healing, and it talks to you about how Pseudomonas affects the inflammatory process and the proliferation process and inhibit wound healing, and how these proteases actually contribute. I would look through this and be familiar with it. I'm not gonna test you too hard on the paper, but if you're interested, definitely look through it and read through it, okay? Okay. We don't have a lot more time to talk about this, so I wanna leave it at that. Actually, I do wanna tell you about one model that can be used to study proteases that's used with Pseudomonas aeruginosa. And this is a mouse model to study wound infections. So this is a mouse, and lots of these studies are done these preliminary studies are done in mice before they're actually done on humans, because there are a lot more problems associated with doing these studies in humans. So Pseudomonas, one of the common animal models is in mice, and what they do to study, to find new drug targets against Pseudomonas is they do these studies in mice. So they have a mouse burn wound model, where they will take a mouse and shave its back, and they cause a burn on the back of the mouse, and then they can infect it with Pseudomonas, different concentrations of Pseudomonas, and monitor the infection process. This doesn't hurt the mouse. The mouse is totally unaware of everything that's going on. It's asleep through all of this. This is just used to monitor the colony-forming units of the bacterium in this mouse model. And so what these researchers have done is they made a number of genetic mutants in Pseudomonas to understand how these factors inhibit wound healing. And that's how they found that the elastase and the elastase proteases contribute to Pseudomonas infections ultimately. And what they found now is that Pseudomonas can also misregulate these host matrix metallic proteins to prevent a cut or a scrape from actually healing. There is quorum sensing. I'm not really going to hit this hard today. There is this chemical sensing process called quorum sensing in bacteria, where different bacterial pathogens or different bacteria actually talk to each other using these chemicals that they secrete. And so this can, when Pseudomonas recognizes a specific chemical signal, it does induce creation of genes that are involved in biofilm formation. So these bacteria are actually talking to each other as well, and that contributes to these skin wound infections and biofilm formation. Yeah, and so just know that elastase and these other proteases can help prevent wound healing by impacting formation of the extracellular matrix and reforming of this connected tissue. Okay, take-home messages from this lecture and the one on Friday. Adhesins are often required for the first step in an infection. If we inhibit a single adhesin in a coordinated fashion, it's often sufficient to attenuate virulence. Therefore, adhesins have been targeted with therapeutics, although none are on the market just yet. Proteases are degradative enzymes, and they're used by bacterial pathogens to initiate and prolong infections. Okay? All right, so any questions of this? All right, email me if you have any questions. I gave you your case studies, the bioterrorism case studies. Hopefully you all had a chance to look over it over the weekend. Each group has a different scenario. So go ahead and discuss with your group and answer the questions on one of the sheets of paper that I included in your packet. I put in a number of different, I think I put in 3 different questionnaires just so that y'all have everybody questions. Okay? But be sure to sign in and give me the questions before you leave today. And we will have time on Tuesday and Friday to do more of this. Spend on bioterrorism case study. We're going to go through it Friday, all the way through it on Friday. We'll do more on